Hi everyone, my name is Emma Schaefer, and today I'm going to be talking about the biosynthesis of cholesterol, what it does for the body, and how high cholesterol is decreased by the drug lovastatin. So first, I'm going to cover why cholesterol is important for biological functions. Cholesterol is important for many reasons. It's a vital component of cell membranes and helps regulate the fluidity of the membrane due to its rigid ring structure. It's also a precursor for steroid hormones, which are important for cell signaling. And then it's also a precursor for bile acids, which helps with digestion. Um, Although cholesterol is important, too much of it can cause problems like cardiovascular disease and strokes as it builds up in the arteries. So, because of this, the synthesis, utilization, and transport of cholesterol must be highly regulated to ensure that there is minimal excess, so there is less buildup. So, now we're going to get into cholesterol synthesis. The starting point for cholesterol is acetyl CoA. Two molecules of acetyl CoA condense to form acetoacetyl CoA. A third molecule of acetyl CoA is added by the enzyme HMG CoA synthase, which creates HMG CoA or hydroxymethylglutarol CoA. HMG CoA is then reduced to mevalonate by HMG CoA reductase. This is then phosphorylated to produce phosphomevalonate, and then phosphorylated again to produce 5-pyrophosphomevalonate. This is then decarboxylated to form isopentyl pyrophosphate, and then four isopentyl pyrophosphate molecules and two dimethyl allyl pyrophosphates condense to form squalene. Squalene then cyclizes to form lanosterol, and then this is then converted to cholesterol. HMG CoA reductase regulates cholesterol synthesis because it catalyzes the rate limiting step of the pathway, which is the reduction of HMG CoA to mevalonate. So, statins are a group of drugs that target HMG CoA reductase by competitive inhibition. Lovastatin specifically is an example of a statin drug. Um, it has an HMG-like group, which is shown in the red images below. So you can see in lovastatin and then also in HMG CoA and mevalonate. Um, it is a competitive inhibitor of HMG CoA and binds very tightly to HMG CoA reductase. It also has bulky hydrophobic groups that HMG CoA reductase must alter its conformation in order to make room for, and this increases the binding. Lovastatin specifically is a lactone, which means it must be hydrolyzed in vivo in order to generate its active form. And statin drugs work very well to inhibit HMG CoA reductase and thus the formation of cholesterol due to their ability to bind so tightly and thus work effectively as a competitive inhibitor.